Hello, my name's Andrew Ward and I'd like to welcome you to this Inside Cancer session on cell signalling, oncogenes and tumor suppressors. What we're going to be talking about is cell communication or cell signalling. And the basic idea here is that cells need to talk to each other in order to coordinate what they do, particularly in any animal, say, which has lots of different cell types doing their specialized jobs. So how this is done is that one cell releases a chemical signal which then interacts with another cell, usually via a specific receptor interaction, so that that cell can respond. Why is it important that cells communicate? Well, like anything that involves lots of interactions, you need to coordinate this behavior. So cells communicate using lots of signals to perform all of the functions that they do. Particularly in multicellular organisms, cells need to communicate for correct development, for instance, during early life, and to maintain tissues and functions later on. So I'll show you a couple of examples of what I mean. So first of all, during development, we all start out as a single cell which has to multiply, and then those cells specialize to become all the different cell types of the body. So if we consider a single cell, it may have a variety of different options that it can adopt. It's surprising, but some cells will be instructed to die. Others require signals to tell them to survive. Those cells that do survive may be instructed to divide, so we can increase cell number. And at some point during the development of an animal, the tissues need to form with their specialized structures and cell types, and that requires cells to specialize or differentiate. And that again requires different signals to ensure that the cells end up in the right place doing the right thing. So if we consider another scenario, which is a fully formed tissue such as skin or epidermis, what we're looking at here is the stack of cells which form the barrier on the outside of the body. And the skin is a continually renewing tissue. And this is because cells are constantly being lost from the surface of the skin, and those have to be replaced, and they're replaced by division of cells in this basal layer at the bottom of the epidermis. So the cells specialize as they move up from the basal layer until they reach the surface, where they're flattened, they're full of a protein called keratin, and they form the waterproof layer that is the skin. So what is needed is tight control over this process of cell division so that there are sufficient cells made to replace those that are lost, but no more. So let's move now inside the cell and ask what happens when the signal reaches the cell surface and interacts with this specific cell surface receptor. What this does is it causes a chain reaction or a relay of molecular events inside the cell, which ultimately allow the cell to change its behavior. So this relay disperses the signal to the parts of the cell where it's needed. So, for instance, the cell may need to release energy to do what it's going to do. The cell may need to change shape. And part of the signal may enter the nucleus, where it can change gene expression so that the cell can change its behavior over the longer term. Again, tight control is needed over the processes that the cells have to undergo, and particularly with reference to cancer, tight control is needed over cell division. One of the hallmarks of cancers are that cells become uncontrolled in their ability to divide. They disobey the normal rules over cell division, and they can divide without instructions. So we're going to look at how this happens. And first of all, we have to consider the life history of the cell. A dividing cell is said to go through the cell cycle. And this is a cycle in which most cells spend most of their time in a resting phase, if you like, called G1 or gap 1. If the cell's going to be instructed to divide, it has to pass from that gap or growth phase into S phase, which is the period during which the cell divides all of the DNA. So S for DNA synthesis. Once the DNA has been divided, then the cell again goes through another gap or growth phase until it's ready to physically divide in M phase or mitosis. So that's the life history of the cell that will go around the cell cycle. Begins at the start, which is between G1 and S phase. That's where the decision's made to begin to divide. But all the way around the cell cycle, there are checkpoints where the cell can make sure that everything is okay to proceed around the next bit of the cell cycle. So we could take a quick look at that. For instance, in G1, that first resting phase, if there's DNA damage, then the cell will not want to go through division and copy that damage into the new cells. If there's an unfavorable cellular environment, not enough energy available, for instance, then the cell will stay where it is. During S phase, if there's incomplete replication of the DNA, again, the cell should not divide because that mistake, if you like, will be carried over into the new cells. During the second gap phase, the cell may need to acquire a minimum size to undergo division. And in M phase, all of the chromosomes have to be correctly aligned so that they can be pulled apart into the two daughter cells correctly. So there are checkpoints at each step which will prevent the cell from going around the cell cycle or proceeding around the cell cycle unless those checkpoints are passed. And the checkpoint that we're going to focus on is the one between G1 and S. So the checkpoint before the cell goes into the DNA synthesis phase. Now remember, the cell signal arrives at the cell surface and interacts with a specific receptor. So that receptor is the one for this particular cell type that will tell it to divide. The signal causes the receptor to trigger a relay of molecular events inside the cell. And these molecular events ultimately lead to this positive decision to divide by transiting the cell from G1 into S phase. And what can happen in cancers is that some of these molecules, one of these molecules perhaps, in the relay, becomes inappropriately activated. And what we can see here is that even in the absence of a signal, this inappropriate activation means that the cell can progress into the first step of the cell cycle. Now, inappropriate activation of these relay components usually happens through gene mutations in cancer. And these mutated genes are known as oncogenes. In addition to these positive instructions for a cell to divide, the cell also has a brake. So it takes both an accelerator and a brake approach to controlling this transit from G1 into S phase. And the components of the system which act as a brake on the cell cycle at this point are called tumor suppressors. So whilst the tumor suppressor is sitting there in the cell, it's stopping the cell from going around the cell cycle. If we again trigger the cell cycle, or attempt to by instructing the cell with a signal, initiating the relay, one of the jobs of that relay is to remove that block, that brake. So you remove the tumor suppressor, and again, the cell is then free to move from G1 into S phase. So another thing that goes wrong in cancers is that these tumor suppressor components, or their genes, become mutated. And if they're mutated and can't work, then again it's easier for the cell to pass through that first part of the cell cycle, even when there's no signal present. So we have both oncogenes and tumor suppressors, which are mutant forms of the normal controls over cell proliferation or division, which you see in cancers. And what we'll see is that cancers then begin as a mutation in a single cell in a tissue. So what I'm illustrating here is a tissue which is just a sheet of cells, and one of those cells acquires a mutation which begins the process of giving this cell a slight advantage over other cells in that it can, in its potential to divide. The cell needs to acquire further mutations in oncogenes and tumor suppressor genes to become overgrown and will first form a characteristic benign growth. But further mutations can allow this growth, or some of the cells, to go beyond simple overgrowth, break through the boundaries of that tissue and start to invade other tissues. And at that point, the cancer becomes dangerous and malignant. So all I want to do to finish is to summarize some of the key things that I've said. First of all, I hope you'll have appreciated that cell signaling is an essential way of ensuring that cell behavior is coordinated and controlled. Secondly, the cell signaling that I've been talking about involves a chemical signal, a specific chemical signal coming from one cell and acting on another. And finally, uncontrolled cell division in cancers is the result of mutations in genes which we call oncogenes and tumor suppressors. Okay, so there we have it, and thank you for joining us today.